Okay, so nervous system, part 2. And here's your data question picture. This picture should look familiar. You should have looked at it last night on the video. Okay, so looking at this question, your data question, somebody raise your hand and tell me which one was not the answer. Let's eliminate one. You think it was really hard? Okay, so somebody tell me A is not the answer. Josh, go ahead, tell me why can we eliminate A?
Because to generate that potential, it's not a massive inward flow of potassium, it's an inward flow of sodium. Perfect. So A is incorrect because it should have said a massive inward flow of sodium. So that actually eliminates 2 of them. Which 2? Is that on the video?
Yeah, so A and B are both wrong choices.
Please tell me. Did you pick D? What'd you pick? Yeah, I did.
Okay, so now we're down to C and D. Okay, the difference in C and D, it says, is it the removal of potassium or is it the entry of a phosphate ion? That's what your question is down to. Is that second part? Because both of them say that depolarization is because of the inward flow of sodium. Y'all see that in the first sentence?
I see that.
Yes, so that is the same on both of them right here.
C, isn't it?
Wait, watch, I can change colors. Ready? I think.
Do it. I don't see it.
Okay, maybe I can't.
LOL.
You have to pay for the $40 app to do that.
Seriously?
That's the light version.
It was 30.
That was $30.
Okay, so on C and D, you've got inward flow of sodium on both of them. So that's the same thing. So that was accurate. Now our choice is it down to the massive removal of potassium on repolarization right here? Is it, on the repolarization, is it flow of sodium? I'm sorry, is it potassium or is it phosphate?
I'm feeling C.
Feeling C. C is the correct answer. D is wrong. Okay, so something also to note. What is wrong with, look, it says on D, the repolarization back to a more negative internal state is occurring by negative phosphate ions entering. So to get more negative, the phosphates aren't moving. What is a phosphate? Come on, somebody make me proud. What's a phosphate?
Adding of a phosphate. Oh, wait, what
Is a phosphate? Okay, it's a phosphate and it's got those oxygens around it, right? It's actually PO4. So phosphate in the middle, oxygens all around it. Okay, where are they located? 2 major places that you know. DNA and ATP, right? Y'all know that? Yeah. ATP has 3 of them. DNA. Okay, so phosphates you want to know are negatively charged. This is just a little reminder. Phosphates are negatively charged. They're in the DNA, and that's what makes the inside of the cell negative. So the inside of the cell has a slightly negative charge if you look here. So looking at this, somebody tell me what this picture shows them. Yeah, we got neuron on the left-hand side. What else? What's on the right-hand side?
Extracellular fluid.
Okay, what is cytosol? Yeah, inside the cell. So this picture is just showing you inside the cell versus outside the cell. So inside the cell, it shows you, y'all see that little negative charge right there? Inside the cell is negatively charged because of the phosphates in DNA. Negatively charged because the phosphates in the DNA. So, and Ryan said the ions. Yeah, there's negatively charged ions in there too. Okay, so from the video last night, you learned that the inside of the cell was negative. But what did he tell you on the neuron? What did he tell you to think of as resting potentials? Anybody remember? How to remember the ions, like where the sodium was and where the potassium was? A salty banana. Does anybody else remember that? Salty banana? Okay, so why did he say that?
Because there's
Potassium's on the inside, good. And salt's on the outside. That's resting potential.
That's how I answered the data.
Yep. Salty banana. Good deal. Okay, so check out your notes for me for just a second. Remember that this is things from you should already know. Energy can be associated with charged particles called ions. How is energy associated with charged particles moving? Somebody give me an example. How is energy associated with charged particles moving? Do what?
Through the membrane, like channels.
Yeah, they're moving through the membrane. Specific example. Come on.
Ions. Gateway.
Okay, let's go with how do we make energy? By moving particles.
Cellular respiration. Breaking bonds.
How do we, active transport requires energy, but how do we make energy by moving particles?
Static electricity.
Think. You know this. How do we make energy? Do what? Yeah. Josh is on it. He said a concentration gradient. How do we make energy, period? How do we make ATP?
ATP synthase.
What'd you say? ATP synthase? How does that work? How does ATP synthase work? It turns, how does it turn? What makes it turn?
Energy.
What makes it turn?
The ions flowing through it.
What kind of ions flowing through it?
Positively charged ions.
Hydrogen ions, right? Okay, remember that those hydrogen ions flow through ATP synthase. So the hydrogen ions are popped up through the electron transport chain, and then they passively go through ATP synthase, and they make it flow. So on the first thing it says, energy can be associated with charged particles. That energy is moving through the, what do we call that whole process? Electron transport chain. So maybe write that in. Y'all should have known that. I'm a little disappointed in that. Okay, it says established, on number 2, established concentration gradients can be a source of energy. What is a concentration gradient? What is a concentration gradient?
Concentration of something.
What'd you say? The movement of the molecules, right? So passive transport is with the gradient or against the gradient? With. Active transport is against the gradient. Do y'all remember that? So active transport flows against the gradient. What that means is it's flowing in the opposite direction of the majority of the molecules moving. Passive transport is flowing with the majority of the molecules moving. And then it says active transport requires a transport protein. Somebody give me an example of an active transport protein. A protein, an example of an active transport protein. What's an example of an active transport protein? A protein, a transport protein. A protein. RNA is a nucleic acid. It's not a protein. An active transport protein. When we talked about passive and active transport, what was an example of active transport? Say it again, Holly. Say it again so everybody can hear you.
Sodium, potassium pump.
Sodium, potassium pump. Y'all knew that, right? Sodium potassium pump is a protein in the cell membrane that actively transports sodium and potassium. Yeah, it is on the board. Little magnets on the board. Okay, so let's review. We need a little review on this transport stuff. What is passive transport? With the gradient. Examples. Come on. Osmosis. Diffusion. One more. Osmosis, diffusion. One more. Facilitated diffusion. Okay, so 3 examples of. Okay. So if you look at the picture, it says membrane potential. This is the ability for the membrane to potentially do work. So it's going to do work or use energy based off these ions. And we actually talked yesterday about anions and cations. Do y'all see those? And you know that anions are negatively charged. Cations are positively charged. Okay, so looking at the picture, somebody tell me. You can see from the picture that the inside's negative, right? Why is it negative? Because of the DNA and the phosphates. Good. Okay, next picture is this is showing you just the sodium potassium pump. So if you look at this purple protein right here, this is the sodium potassium pump, and it's going to open and close. You see how it's like getting ready to pump those sodiums out? Okay, so what the sodium potassium pump does is it maintains resting potential. Resting potential is where a neuron is not sending an impulse. It's an unstimulated neuron. So resting potential is unstimulated neuron, not sending an impulse. Sodium-potassium pump maintains it. So when is the sodium-potassium pump used?
After repolarization.
Yeah, to maintain resting potential, and Ryan said after is repolarized. That works. Okay, so sodium-potassium pump. Pump sodium out, potassium in. How can you remember that? Salty banana, right? It's trying to maintain the salty banana. So it's trying to maintain that inactive neuron. And then it says it's a membrane-bound protein. So it's in the cell membrane. In the cell membrane here. And you can see, look, it's picking up sodiums from inside the cell, and it's pumping them out. Because the salt goes on the outside. It's picking up potassiums and pumping potassium in. Okay, so if you think about a neuron, bet you see a neuron here. Maybe. Yeah, you do. Oh no. Okay, so if you see a neuron, here's the cell body. There's the dendrites. Do y'all know what they do? Dendrites receive. Cell body does what? Interprets it and changes it. It does transduction. And then the axon goes down. Okay, so on the cell body, so look at my picture here. On the axon, on the cell membrane, is that sodium-potassium pump. So there it is. It's going to send sodium out and potassium in. Oh, that's just a K, not a Ka. So sodium out, potassium in. But I drew this so you can see that it's actually on the cell membrane of the axon. Okay. So gated ion channels, you already know what that is from the last test. Those are the remember the blue magnet that had the little gate on the top? Do I need to pull up my magnet? Don't make me pull up my magnet. Y'all got it? Okay, so those are the gated ion channels. Remember when the ligand binds, the gate opens, and what can flow through? Ions can flow through. Okay, so there's specific gates, and this one is showing one for sodium. Oh, I'm having a hard time today. Oh. Okay, so the first one right here is showing, I don't know. Okay, so a gate for sodium right here. And do you see how the little gate is open? And then the sodium can flow? Can y'all see that? And then on the potassium one, the potassium can't go in. It goes out because the little gate is closed. And then the sodium one here is closed. Can y'all see that little bitty gate? Right there is closed, and the potassium one is open so it can go in. So just like the magnets I showed you last time. When the ligand binds, the gate opens, the sodium can flood in, the sodium can flood out. But there's specific ones just for sodium, and there's specific ones just for potassium. And they're used for the depolarization and repolarization. So the sodium-potassium pump is used to maintain resting. The channels are used to depolarize and repolarize. Okay, so on gated ion channels, there's two different ones. There's ones that are used chemically with ligands. That's like the blue magnet from the last test. The ligand opened it up. Okay, and then the voltage gated ion channel opens up because of charge. So chemically gated ions open up because of a ligand. And then voltage ones open up because of the charge. Okay, so go ahead on depolarization. I hope you know this, but sodium floods in. Highlight that. So if you think of resting potential as polarized, in order to get out of rest, you've got to depolarize. Remove that. Okay, and this depolarization is when the sodium comes in, and there's a threshold, and that's like enough sodiums come in that the action is going to go down. So threshold is like, I want to call it the point of no return, kind of like the checkpoint of mitosis. So enough sodium flooded in that the action is going to go down. What might the action be? What might the action be? Flexing a muscle, making a gland release a hormone, a movement, something like that, right? So you've got to get the chemical from your brain down the axon. Okay, so go ahead, and then action potential is actually when the message is sent. So electrical generated impulse in a nerve. So if everybody will, one person from each table, come get one of these blue sheets of paper, and then come get a baggie of proteins and ions. Okay, so look at this picture. Make sure you can kind of see it with this picture. Somebody tell me what happened during depolarization. What molecule moved? Sodium went. Sodium. Which way was it? Sodium flooded which way? Well, if it was salty banana, now the salt is on the inside. The sodium is on the inside. So depolarization means sodium floods in through the channel, through the channel. So, okay, depolarization, sodium floods in through the channel. Okay, action potential, that's where the action actually goes. The message was sent down the axon. So it got to do y'all see the plus 30 charge right there? Yeah. It's got to get to that plus 30 charge, and then it goes. Then there's repolarization. What happened then? Potassium shot out through the channels, right? Okay, and then what was it right here? What was that called? Hyperpolarization, where too much potassium went out. Yes? Okay, and then back to resting potential. What maintains resting potential? Sodium potassium pump. So look at these numbers. This is something we didn't look at yet. If you look at the numbers, so resting potential is negative 70. So resting potential is negative 70, sending sodium out, potassium in. Okay, depolarization, sodium rushed in until it got to action potential. That means the action goes. Repolarization, potassium out, too much potassium, and then back to hyper. One more word from this graph. It's actually not on this graph. But if this is negative 70 and that's positive 30, when it gets from negative 70 to about negative 55, right there, that's like the point of no return. That's what we call the threshold. So the threshold is like if it gets to negative, if it goes positive to negative 55, because it was negative 70. Y'all see that? If it gets to negative 55, it's going. The action is going to go. But it doesn't go until it gets all the way to positive 30. Does that make sense? Yes, that makes sense? Okay, so I actually have a video for you to watch.
Did you make it?
No, last period did.
Last period made it or stuff?
No. Not really good, huh? Okay, so sodium flows in, that's depolarization. I can just use the words myself. Okay, so this is repolarization. Potassium comes out. Potassium comes out. Potassium comes out. Okay, too much would be hyperpolarization. And then they would use the sodium potassium pump somehow soon. There we go, to get it back to resting potential. Okay, so what I want you to do is I want you to try it. Like you practice until you've got all. Okay, so just kind of at your tables, discuss what's happening at each of these things. Notice the charge, because that's important. Notice the charge. discuss what's happening at each part.
Depolarization.
Wait, repolarization.
So is it going to be at negative 55 every time?
Negative 55 is that threshold.
That's every time?
That's like the point of no return. That's all or nothing.
So it's always at negative 70 and positive 30?
Yeah, negative 7 is resting, and then positive 30 is go time.
Are there any like math questions to do with this? Do you have any math questions?
Okay, one thing, make sure you know what that threshold is, that point of no return. Make sure you discuss that too. 55. Yep.
Depolarization. Yeah, NA goes in. And then repolarization, K goes out. Too much goes out. And they go through the potassium. Potassium comes to the pump and sodium. No, they both go through.
Okay, so if you'll come back to your notes real quick. Instead of saying the nerve impulse reaches that positive 30, instead we call it propagation. So this is when the impulse travels down the axon. So instead of saying the impulse travels down the axon, we call that propagation. But one thing to kind of look at, the way we did it on our little pieces of paper, we did all the sodium in, all the potassium out. But really what's happening, okay, so the impulse is traveling down, and so right there where it's blue, the sodium is flooding in. Okay, well, so as the impulse continues to go, so now where that red line is, the sodium is still coming in, but behind it, the potassium's going out. So it's like the back of the, well, I guess the front of the neuron. The other side of the neuron, right here, is repolarizing as it's still going down. Does that make sense? So now when it goes a little bit further, where this red line is, right here, so now the sodium's flooding in, but right here, the potassium's moving behind it. So it's like happening all at one time. Does that make sense? So as the nerve impulse goes down, it's depolarized, and then right behind the nerve impulse, it's repolarized. Does that make sense, the way I've said that, looking at the picture? Okay, we said yesterday, the nerve impulse, it doesn't go straight down. How does it go? It jumps, and that is called, instead of calling it jumps, it's called saltatory conduction. Oh. Okay, so it jumps like, I want to think like a frog on a lily pad, but it jumps in between those Schwann cells, and you probably saw from the video last night, it jumps in the gap right here, not the gap like the synapse, but the gap in between the Schwann cells, right there. It's called nose of Ranvier. Do y'all remember seeing that on the video? So it jumps in between those little Schwann cells. And Schwann cells make up the myelin sheath. Good. Okay, last thing on today's notes. The reflex arc. You know when you go to the doctor and you get your reflexes checked? Okay, what they're really checking is your homeostasis. They're checking, by checking your knee, they can actually see if your temperature would rise if you had an infection, or if your heart would increase if you reached adrenaline. So they're checking all of your reflexes, and if that one works, most of them do. Does that make sense? Yeah. Okay, so your reflexes, what it is when they hit your knee at the doctor, the sensory neurons right at the knee send the impulse right to the spinal cord and then right back. So you never even think about a reflex. Like your knee pops up, you don't have time to think about it, right? Yes? Okay, so it's because the sensory neuron sends the message to the spinal cord and back. It doesn't ever go to your brain. You don't ever have to think about it. So it goes sensory neuron, and then interneuron neurons in the central nervous system. Central nervous system, remember, our brain and spinal cord. And then it goes back to the motor neuron. And what is the motor neuron? That's going to act on the muscle. That's the neuron that's going to act on the muscle, right? So there's 3 kinds of neurons that you want to know. Sensory neuron, where are those located?
In your brain.
Sensory neuron. They accept the sense. So they're the touch, the hear, the taste, the smell, right? They receive the senses. Interneuron, those are in the brain. And then motor neuron, where are those at? Muscles. They act on the muscles. They act on the glands. So you want to, real quick, think about those 3 types of neurons. You want to know those. Sensory neuron are ones that receive the message. Interneuron, those are neurons in the central nervous system. With your reflex arc, they're in the spinal cord. And then your motor neurons, they act on the effector cells. And the effector cells are either muscles or glands. All right, any questions? Yes, sir.
What happens if you don't have a reflex in your knee?
Well, I think it doesn't matter about your knee, but what it is the reflex is like, okay, automatically when you get sick, you get a fever. That's a reflex. When adrenaline is released, your heart speeds up. So by checking your knee, they're really checking all your reflexes. So I guess if you don't have reflexes in your knees, then they would have to do further tests to see what the problem is. Good question. Any other questions? Do what now? I don't know. I like that. That's why they check your reflexes when you go to get like a physical test, like to play sports or whatever, to see if your reflexes work. Because if they don't, then they need to do further testing to see what's up. Okay, get out a sheet of paper for me. Synthesis question. Do what now? No, but that would be a good one. I hope you could write about that. Ryan said, is it going to be 3 types of neurons? I hope you could write about that if it was. It's not. Okay, so put your notes away for me and answer this question. Get this answer. So you should have had depolarization, sodium entering, repolarization, potassium out, hyperpolarization involving potassium being reabsorbed. And if y'all will actually grade these two.